Hello and welcome to continuation of Heart 2. This is cardiac muscle tissue. So in this cartoon you can see we have a muscle cell. You can tell it's a muscle cell because you can see the striations or the bands of actin and myosin in the sarcomeres. So if you listen to the previous podcast, which is skeletal muscle review, you'll recall that myofibrils, which are these long cylindrical structures in this cartoon, myofibrils, are made up of sarcomeres. And the sarcomeres are the contractile units of both skeletal and cardiac muscle cells. So in this cartoon, if you look at the bottom muscle cell, you can see sort of a zigzag blue line. That's a Z disc, and so if you go from Z disc to Z disc, that's one sarcomere. And so when the Z discs get closer together, that's going to ultimately cause your muscle to shorten. So if, for instance, if your bicep muscle is made up of millions of these sarcomeres within the muscle fiber, then when the sarcomeres get shorter, that's going to cause muscle contraction. So it's the same thing with cardiac muscle. Cardiac muscle has actin and myosin filaments that slide past each other when the myosin head binds to the myosin binding site on the actin filament. And then the myosin head moves and causes these Z discs to come closer together, thus shortening the sarcomere, thus causing contraction of the heart muscle so that you can pump blood. So in this cartoon, you can see the orange lines. Those are the myosin filaments, and then the light blue lines that are attached at Z discs, those are the actin filaments. So the myosin filaments are thinner and the actin filaments are thinner. And then another thing you can notice on this cartoon, there's mitochondria. There's a lot of mitochondria in heart muscle cells because you need a lot of ATP to constantly be contracting your heart muscle. So, for instance, your skeletal muscle, your bicep, isn't always contracting, but your heart muscle cells are always contracting. So they need a lot of ATP, so they need a lot of mitochondria. The purple discs in the cartoon is the nucleus. It's in cardiac muscle cell. There's one nucleus. In skeletal muscle cells, there's many nuclei. There's also a sarcolemma around the cardiac muscle cell. And remember from the previous podcast on skeletal muscle, the sarcolemma is just the plasma membrane of the cell. So here's our cartoon again, and then at the bottom of the on the right side, the lower cartoon actually shows you the I bands and the A bands and the lines and the discs of a sarcomere. So if you look at that cartoon, you can see that, like I said in the previous slide, the myosin filaments are those orange lines. The thin filaments are made up of the protein actin, and they're the thin blue lines. There's an A band, and if you look at the A band, you can see the A band consists of overlapping actin and myosin filaments. The I band just contains the region where the Z disc is and part of the actin. The H zone contains an M line where the myosin filaments insert on the M line. So, like skeletal muscle, cardiac muscle is striated, and what that means is the actin and myosin myofilaments overlap. So you can see in the cartoon how they overlap. Unlike skeletal muscle, however, the cardiac cells, or they're also called cardiac myocytes, they're shorter than a skeletal muscle cell. They're thicker, cardiac muscle cells are thicker than skeletal muscle cells, and they branch. So you can see in the cartoon, in the upper part on the right hand side, you can see how you have one cardiac muscle cell at the top, and then you have a little branch point onto another cardiac muscle cell. And they also just have one nucleus, and I said that earlier, as opposed to skeletal muscle that is multinucleate. There's limited mitosis of these cardiac muscle cells, so there's a decreased ability to repair your cardiac muscle tissue or your heart when it gets damaged. There may be satellite or stem cells to do that repair, some limited repair, but mostly when you damage your heart muscle cells, the fibroblasts that live there will just create scar tissue in the heart, so then the heart won't pump as well. Oh, and then one other thing I forgot to mention, in the cardiac muscle cell, there is a structure called myoglobin. So myoglobin stores oxygen. So that's why heart muscle is so red. So if you remember when we looked at the beef heart in class, how really dark red it was, that's the myoglobin that's storing the oxygen, so that your heart will never be out of oxygen. Your heart muscle cells will never be low on oxygen, so they'll be able to beat for as long as you live. There's also glycogen, a lot of glycogen in your heart muscle cells for glucose storage, so you can break down glucose to make ATP. Unlike skeletal muscle, cardiac muscle cells, aka cardiocytes, also they have less sarcoplasmic reticulum, so there's less calcium storage in cardiac muscle cells. But like skeletal muscle, the cardiac muscle will allow calcium influx into the cytoplasm of the cardiac muscle cell from the sarcoplasmic reticulum, which in the cartoon is that blue sort of network of tubes inside the cardiac muscle cell. In cardiac muscle cells, the T-tubule, which is that part of the plasma membrane that dives into the inside of the cardiac muscle cell, is a little bit wider in cardiac muscle cells. And like we said on the previous slide, there's a lot of large mitochondria in cardiac muscle cells for aerobic cell respiration. So if you don't remember what aerobic cell respiration is, then you can ask me in class, or you can go to your 156 textbook and read up on that. About 25% of the cytoplasm in cardiac muscle cells is occupied by mitochondria, and about 2% of the skeletal muscle cytoplasm has mitochondria. So the heart muscle cells can use alternative fuels. So the main fuel we've talked about for cells like to eat glucose, but they can also eat or break down fatty acids or proteins, and the heart muscle cells can use these alternative fuels like fatty acids or proteins, and they can switch their metabolic path from using glucose to using fatty acids pretty easily. So they'll never, well, I shouldn't say never, but it's hard for them to run out of fuel because they can use different fuel sources. So this is a cartoon showing two cardiac muscle cells where they come together in this thing called the intercalated disc. So you can see in this cartoon there's a sarcolemma. Remember, that's just the plasma membrane of the cardiac muscle cell. There's mitochondria. There's a single nucleus per cell. And then within the intercalated disc, if you look at those purple arrows that show you up close what's in an intercalated disc, there's a structure called a desmosome, and that's just sort of a weld or a patch between two cardiac myocytes, and it's made of protein, so that it can sort of weld the two plasma membranes together. So that's more of a the desmosome is a structure that holds the two cells together, as opposed to the gap junctions. So if you can see that on the right of the cartoon, there's gap junctions. They're used so that one cardiac muscle cell can electrically signal the cardiac muscle cell right next to it. So desmosomes are more for structural attachment, and gap junctions are for electrical signals between cardiac myocytes. There's a lot of surface areas. You can see the way the intercalated disc is structured that gives the plasma membrane a lot of surface area, so that there's a nice close fit between two adjoining cardiac muscle cells. So we talked about the desmosomes. There's also, in addition to the desmosomes, it doesn't show it in this cartoon, but there's another protein structure called the fascia adherens, and that anchors the actin of one cardiac myocyte to the actin of another cardiac myocyte. So, but the fascia adherens also prevent cells from breaking apart. And then the gap junctions electrically connect each other so that the cardiac cells act as syncytium. And what that means is the atria can all the cardiac muscle cells of the atria can contract at once. All the cardiac muscle cells of the ventricle can contract at once, because all the cells in the syncytium get this electrical signal to contract at virtually the same time. And this is just a cartoon. We looked at this in the skeletal muscle review, but if we start at step 1, we know there's calcium present because the troponin tropomyosin complex, which is that little yellow knob on that ropey orange thing, that's troponin tropomyosin complex, has moved out of the way of the myosin head binding sites on actin. So the actin are those the blue or purple balls with the little black hole in the middle of them, and then the orange structure is myosin head. So ATP has been hydrolyzed to ADP and inorganic phosphate, and the myosin head is bound to the myosin head binding site on actin. In step 2, when ADP and inorganic phosphate are released from the myosin head, that means the myosin head can move, and it pivots toward the middle. And when you get a million sarcomeres doing this at once, you're actually going to shorten your sarcomeres, so the Z discs are going to get closer together, and the entire muscle will shorten. Then if ATP is present, ATP will bind to the myosin head and release the myosin head from the myosin head binding site on actin. And then ATP will hydrolyze, the myosin head will get back into the cocked position, and it'll be ready to cycle through another binding and moving the sarcomere. So this is just a close-up of what's going on when an electrical signal comes into the cardiac myocyte. So you can find your sarcolemma. So find the sarcolemma on the cartoon. That's just the plasma membrane of the cardiac myocyte. And then you can find the T-tubule. And if you follow down inside the T-tubule, you'll see these voltage-gated calcium channels. So when an electrical signal, like an action potential, travels down into the T-tubule, because it's an electrical signal, then the voltage-gated calcium channels will respond to that electrical signal, and they will cause release of calcium from the sarcoplasmic reticulum. And when calcium is released into the sarcoplasmic reticulum, hopefully you know the rest of the story. Calcium can bind the troponin tropomyosin complex and move actin out of the way of the myosin head binding sites on actin, and then the myosin heads are free to bind and pivot, and you can shorten the sarcomere and thus contract the muscle. And I think that's it.